In the world of science, there are two separate but equally important types of cell division: mitosis, which divides a cell into two identical daughter cells, and meiosis, which divides a cell into four unique haploid cells. These are their stories.
Excuse me, sir, are you with the Ann Arbor Police Department?
Not anymore. I work alone now.
You're still a detective, though, right?
Yep.
I have something I need to be solved. I've got the money, I just need the expertise.
What's the mission?
Mitosis versus meiosis.
I know just whom to talk to.
Is that even the proper form of whom?
I try to get out, but they just keep pulling me back in.
Doc, I need your expertise on this one.
Fine, what's the mission?
First, we gotta lay out the steps of mitosis, how somatic cells divide.
I'm a scientist, man. I know what mitosis and meiosis are already.
Just investigate with me.
I mean, that's fine, but I could just tell you right now.
Don't be lame, man. Just humor me. Yeah. Indigo, it's me.
Detective!
We've got it. We've got mitosis. We've solved the mission.
Actually, that's only what somatic cells go through, so we only have half the mission done, so yeah.
I always do this, man. It's always one-upping me, and just 'cause you, went to school all 12 years, and you just think you're better than me.
Sorry, man. I shouldn't have said that.
It's okay. Indigo, you still there? We'll be there in a second. First is interphase. Technically, this is not a phase of mitosis, but it is key in preparing the cell for mitosis. It provides time for the cell to grow, perform normal functions, and duplicate DNA. This is what a cell looks like after interphase. The first actual phase of mitosis is prophase. In prophase, the nucleus fades. Then chromatin condenses into chromosomes, centrioles go to opposite sides of the cell, and spindle fibers form. The second phase of mitosis is metaphase. During metaphase, spindle fibers attach onto chromosomes and line them along the center of the cell. This is what the cell would look like after metaphase. In the third phase, anaphase, spindle fibers pull chromosomes apart, and chromatids move towards opposite sides of the cell. In the fourth and final phase, called telophase, chromosomes arrive at the poles of the cell, nuclear membranes start to form, and spindle fibers fall away. This is what it looks like at the end of telophase. To fully form two new daughter cells, the cell must divide the contents of its cytoplasm in cytokinesis. And now, at the end of mitosis, you have two new daughter cells.
Oh, now I get it! Now we have to figure out meiosis.
Yeah, good call, man.
Come on, man. Caillou? Trying to figure out meiosis. I'd be professional for once. How's it going, man? How you doing? Pretty good? Pretty good. Alright, cool. So before we lay out the steps of meiosis, there are a few things you need to know about meiosis as it relates to mitosis. Meiosis creates 4 haploid cells from one diploid cell. Also, meiosis occurs in germ cells, in cells that will eventually turn into gametes, or sex cells. Meiosis can be divided into 2 groups, meiosis 1 and meiosis 2. So first, let's go over the steps to meiosis 1. In prophase 1, the nucleus breaks down, spindle fibers form, Centrioles move to opposite sides of the cell, and chromatin condenses into chromosomes. Quick note about something that happens in prophase 1 that's unique to meiosis: crossing over. Crossing over is when a portion of one chromosome is exchanged with the corresponding portion of the other homologous chromosome. This happens to make sure that all cells have genetic variation. Now, in metaphase 1, homologous chromosomes are lined up on the equator of the cell. They line up in random order to have genetic variation. Now, in anaphase 1, chromosomes are pulled apart from each other by the spindle fibers. Homologous chromosomes are pulled apart, but chromatids stay attached together in the chromosomes. Only chromosomes are pulled apart from each other. Now, in telophase 1, a nucleus forms, spindle fibers disassemble, and now cytokinesis happens and we have two haploid daughter cells. Meiosis 2 separates sister chromatids, and at the end of meiosis 2, we have 4 haploid daughter cells.
Go.
Prophase 2 is the same as prophase 1, except chromosomes don't condense. So first, the nucleus breaks down, centrioles move to opposite poles of the cell, and spindle fibers form. In metaphase 2, spindle fibers line up the chromosomes along the equator of the cell. In anaphase 2, sister chromatids are pulled apart. Now, in telophase 2, the nucleus forms, and spindle fibers disassemble. Finally, cytokinesis occurs, and we have 4 haploid cells formed from one diploid cell. And these are your 4 haploid daughter cells.
Now I'm gonna take over the world!
How? You just know how meiosis and mitosis work.
Yeah, Indigo, whenever we're trying to teach you something, you always do this. It's like whenever we're trying to just have a good time, you just want to take over the world. Really, it ruins the fun. You know, let's go, Ben.
No.
I'm so alone.